So good morning, everyone, and thank you, Jenny, for the introduction and for the invitation to speak with you, and thanks to all of you for coming out on a Saturday to listen and hear about multiple myeloma. I'm going to be speaking about my favorite topic, which is CAR T cells. Jenny asked me to not just talk about CAR Ts for myeloma, but to give a little history about the development of CAR T cells, and a little bit about Penn's role in that development, because a lot of what's out there now was based on some research that was done at Penn, so I'll try to go through that story. But first, we're going to go back to school a little bit and do a little Immunology 101, just to help us understand when we talk about CAR T cells, what actually are these cells that we're talking about. So we have multiple different types of immune cells circulating in our bloodstream. We talk about, let's see if this has a pointer. No, that was the wrong thing, sorry. On the left side of the slide are what's called the myeloid cells, and the main ones you probably are familiar with are called neutrophils, which are a part of our white blood cells, very important as a first responder to bacteria and fungi, for instance. On the right side are the lymphocytes, or lymphoid cells. And these are divided into two main groups: the B cells, which ultimately give rise to plasma cells and make antibodies. Antibodies circulate in our bloodstream, can last for long periods of time, and can help us sort of recognize bacteria and other pathogens that are floating around in our bloodstream. And then what we're going to talk about are T cells. T cells are very important also in immune-fighting, infection-fighting cells. They recognize cells that have been altered in some way by a virus or some invading pathogen and eliminate those cells. And these cells have memory. They can actually last in our bloodstream for years, and so they were a good sort of source of potential cells to fight cancer. And it turns out that T cells have the ability to recognize and kill cancer cells. We've known this for quite some time, that you can actually take a T cell and get it to recognize a cancer cell. And in fact, I can't show the video here, but the bottom shows a video of a little T cell that I'm pointing to that takes on one of those cancer cells and actually divides and kills those cells. But there also are many cells, which you can't see, unfortunately, in the that sit around. The T cells don't interact with the cancer cells. They're sort of inert. And that's because, unfortunately, over time, the cancer cells learn to evade the T cells, or the T cells become what's called exhausted. They sort of poop out. They lose the ability to fight, and they're no longer able to kill. And so a big effort over the last 20 years in cancer therapy is how can we re-engineer or energize those T cells to now become effective cancer killers again. And in solid tumors like breast cancer or lung cancer or melanoma, kidney cancer, there's something called checkpoint inhibitors, these drugs called Opdivo or Keytruda you've probably seen ads for. Now those don't work quite as well in the blood cancers, but what has worked in blood and bone marrow cancers are cellular therapy like CAR T cells, which is what we're going to focus on, and then another group called bispecific antibodies or bispecific T cell engagers, which Dr. Garfall is going to talk about this afternoon. And both of these are ways to sort of wake up your T cells to attack cancer. But the ones that came first were called CAR T cells. So what is a CAR? CAR stands for chimeric antigen receptor. And if you remember your Greek mythology, the chimera was a mythical creature with the head of a lion, the body of a goat, and tail of a serpent. And so a CAR is actually a receptor that does not exist in nature. It's been engineered to have the head of an antibody so that that sticks out on the outside of the T cell, and then has the body and tail of a T cell signaling apparatus. And what it allows you to do is re-engineer that T cell to, instead of recognizing a bacteria or a virus, to now recognize a cancer cell. And it has unique specificity, so that what's called the antigen binding domain, that the part at the end that sticks out recognizes an antigen, which is just a marker on the surface of a cancer cell. And so now you can make your T cells recognize whatever you want, as long as you have a good target that exists on your cancer cell. And this actually was not a new concept. This was first described in the late 1980s by an Israeli scientist named Zev Eshar, but it took over 20 years to really develop this in a way that it could be made repeatedly and reproducibly and get into patients. And one of the key advances was what's called the co-stimulatory domain, that little orange part on the inside of the cell that allows the cells not to die off right away when they recognize the antigen. The cells can survive and grow and divide. And so this concept of a CAR, after 20 years of development, was finally able to get into patients, and I'll show you some of that data. Now, you need to have a target that is going to work, that's on your cancer cells, but is not on a large number of other cells in the body. Otherwise, the T cells are just going to go haywire and start attacking your own good cells. And so one of the first targets that was identified was CD19. And this was a very attractive target because it exists on B cells, which are the cells that give rise to a number of blood cancers like B-cell non-Hodgkin's lymphoma, CLL, and ALL, acute lymphoblastic leukemia, but it wasn't expressed on any other cells in the body. So this was a good one to go after. And then what we'll hear about later is one called BCMA, which is the one you're probably familiar with. That is the target for a lot of the myeloma therapies. But first, how do you make a CAR T cell? And so one of the problems to overcome is you had to get the DNA that encodes that new CAR into the T cell and get it to be expressed. And so scientists came up with this really novel way is to utilize this lentivirus. A lentivirus is a virus like HIV that is very good at infecting T cells. And they took out all of the guts of the virus so that it wasn't able to replicate or cause disease, but they put in the genetic material that encoded the CAR. And so then you can take T cells in a petri dish or test tube, you can infect it with this virus that encodes the genetic material for the CAR, and now that T cell starts making that CAR to large numbers on the surface of it. And then when you put it in against a tumor cell, you can actually kill the tumor cell, and it turns into a dead tumor cell, just like this. And so this has three features: cytotoxicity, it's able to kill; cytokine production. Cytokines are molecules that actually sort of regulate the immune response and tell other cells to start to divide and grow. And then long-term persistence, that's the important thing. These are made from the patient's own cells, so they're not rejected. The cells stay and can actually survey against the tumor for months, and in some cases, they've been found years later in patients. And so this is a living drug that's made from your own cells that stays in your body and attacks cancer. And so how does this work in practice? So patients undergo something called leukapheresis. If you ever had a stem cell transplant, you're probably familiar with stem cell collection. It's the same process. You're on this machine that looks like a dialysis machine. Some blood goes out, they suck off some of the T cells, and the blood goes back in. Those T cells are then sent to a manufacturing facility where they're activated, they're transduced to express the CAR, and then they're grown up into large numbers. And this was actually one of the rate-limiting steps. It took years to really develop this. And actually, Bruce Levine at Penn was one of the sort of leaders who helped figure out how to do this and do it in a safe and controlled fashion, in a sterile way, so that at the end you're able to get the cells out in much larger numbers. And this process can take anywhere from 2 to 6 weeks, depending on the product. And then their T cells are shipped back to the treatment center where they're infused into the patient, usually after 3 days of low-dose chemotherapy that temporarily lowers the white blood cells to make room for the CARs to go in and expand. And so this is the process of CAR T cell therapy that goes on today. And so back in around 2009, 2010, a number of scientists at Penn were sort of getting together and had developed this and were ready to take it into patients. And really, we had no idea if this was going to work. And one of the leaders of this whole process and one of the fathers of CAR T cells is Carl June, who's there in the middle there, and was the scientist who really helped develop a lot of these breakthroughs for CARs. And then on the far left is Bruce Levine, who at Penn helped figure out the manufacturing process. And on the far right is David Porter, who was the head of our bone marrow transplant group at the time and wrote the initial trial. And this is with one of the first patients on the trial. And so the first disease that this went into was called CLL, chronic lymphocytic leukemia. This is another blood and bone marrow cancer. And back in 2010, this could be treated, and there were treatment options with chemo and other agents. But once you became resistant to those, there really were no good options for this. And this was the very first patient, August 2010, who went on the trial, had 7 prior therapies, had no standard options available, got 3 days of low-dose chemo called fludarabine and cyclophosphamide, and then got this CAR product, which was called CTL019. And about 10 days after the infusion, started to get very sick, high fevers, starting to get low blood pressure, difficulty breathing. And at the time, no one had really seen this before and didn't know what this was. We now know, in retrospect, this is what's called cytokine release syndrome. And I'll talk a little bit about this at the end. This is sort of the most common toxicity of CARs. But he actually got very ill, required ICU-level support, but then started to get better after a couple of weeks. And remarkably, all of his leukemia had disappeared. It was almost like a miracle. His bone marrow had been packed with this leukemia cells that was completely empty. His blood counts normalized. He had bulky and large lymph nodes filled with leukemia cells. They all went away. And he went into this complete remission. The CAR T cells expanded more than 1,000-fold in his bloodstream. And actually, he had a complete remission that lasted over 10 years and still had some of the CAR T cells detectable. It was really a remarkable sort of first patient experience. And he actually decided to buy this tricked-out RV and spend the next decade sort of traveling around the US. He shared these pictures and has the fireplace and everything. So it was quite an amazing story for a patient who didn't really have any other options at that time. And it really showed that this could work. And so that initial study, they only had funding for 3 patients. And 2 out of those 3 had complete responses that lasted for many, many years. One had a partial response, unfortunately, that relapsed within several months. But this is a calculation of the sort of mass of tumor when they calculated all their lymph nodes and sort of added it up. And these were pounds of leukemia cells that ultimately went away with these CARs, showing the power of this approach. And this led to the first publication really showing this success by Dr. Porter and colleagues in the New England Journal in 2011, and a companion piece in Science Translational Medicine showing that this could work. Now, around the same time, 2011, 2012, there was also interest in saying, can we apply this in other diseases that express CD19? And so ALL, or acute lymphoblastic leukemia, is a very aggressive bone marrow cancer. In children, pediatrics, it actually has about an 80% cure rate with standard chemotherapy. But for that 20% who don't get cured, it can be very tough to treat. And often patients get a bone marrow transplant that doesn't work, and median survival, as you can see, can be very short. So there was a need for patients with advanced ALL, particularly in kids. And so Stephen Grupp was a pediatric oncologist at CHOP and was working again with Carl June's group to take this same CAR T cell called CTL019 and look at it in patients with ALL. And the very first kid who was treated on this trial, her name was Emily Whitehead. And she was 7 years old at the time. She had progressed after multiple treatments, including stem cell transplant. And unfortunately, she had run out of options, and she was the first patient who got treated. And she actually got very sick, similar. She had this cytokine release syndrome. And this was the first case of using a drug called tocilizumab, which is an antibody that sort of blocks some of these cytokines and calms down the immune response. And she also had this dramatic response and went into a complete remission. And there's this very amazing short video or film. It's about 3.5 minutes long that I think I'm going to show in the breakout session, since I don't have the ability to do it right now. But if you get the slides, it's worth checking out because it sort of talks about her story and the docs who did this. And now she's literally become the poster child for CAR T cell therapy. She and her family have started a foundation to help raise money for patients getting through CAR T cells and for research. And she's now actually more than, I think, 13 years still in remission. She's actually an undergraduate at the University of Pennsylvania right now. So sort of coming full circle, which is quite amazing. So it shows the power of this approach. And this led, actually, that study led to the first gene therapy for cancer called Tisagen-Lec-Lucel, or Kymriah. And this was based on that same CAR that was developed at Penn. And this is sort of a mob that, I guess, gathered. I'm somewhere in there in the middle. But to sort of celebrate this when this was announced in 2017. Now, I've been giving you a very Philly-centric view of this. Penn was, of course, not the only site that was developing CAR T cells. There actually were trials going on in parallel from other great researchers and scientists at Sloan Kettering, at the NIH, and at Fred Hutch in Seattle. All of these really showing proof of concept that you could use CAR T cells to attack these B-cell cancers. And some of these could be durable even more than 10 years later. And so we now have 4 FDA-approved CAR T cell products for these other cancers. And I've listed these here, and these have become part of the standard armamentarium. But what about myeloma? We're, of course, here to learn about myeloma. And so at Penn around 2014, as we were getting the initial successes in these other diseases, there was an interest in saying, can we use this in multiple myeloma? And we knew that CD19 was not a great target for myeloma. It's expressed on a subset of cells. But there was this idea that maybe it was on what's called a myeloma stem cell, a cell that was a precursor that gave rise to myeloma. And so back around 2013, 2014, Al Garfall, who you'll hear from later, one of my colleagues at Penn, and Ed Statmauer, designed this trial to use the same CAR T cells, because we happen to have them available, and say, is there a way we can combine it with standard myeloma treatment? And so this was a trial for patients who had already had a prior stem cell transplant for their myeloma. The disease came back, and they were getting another transplant. And usually when you do a second transplant, it doesn't last as long, the remissions. And so the thought was, I'll do the second transplant, and then I'll give them these CD19 CAR T cells. Maybe that will help extend the time in remission. And again, it's always the first patient. The first patient who got this, you can see on the bottom, that's showing her IgA levels in M-spike. Her IgA was like 5,000, 6,000, and she was needing infusions of Cytoxan to keep her disease barely in check. She got melphalan, which is the drug we give for a stem cell transplant. She got the CTLO-19 CARs, and her disease went away. She went into this complete remission. And it lasted, her first transplant only lasted 6 months, and this one had lasted over a year when we first reported this data in the New England Journal and had not come back. And this got published in the New England Journal. She actually got on the cover of Parade magazine, and there was some press about this, and they talked about a potential cure for myeloma. Now, unfortunately, this was not a cure. Her disease did eventually relapse, but it came back in a sort of less aggressive form, and by that time, there were new drugs that had been approved. Daratumumab was not approved yet when she was treated, and then she was able to get that and other drugs, and she's able to still be alive today now that she's made it sort of to these other medications that were available. So CD19 CARs for myeloma were successful, I think, in this case, but in the trial, they enrolled 10 patients, and only 2 really seemed to have much benefit. So the thought was CD19 may not be the best target for myeloma. There may be a subset that benefit, but we need something better that's on the bulk of the myeloma cells. And so that led to the exploration of a target called BCMA, or B-cell maturation antigen. And this is a receptor that's on all of our normal plasma cells. Plasma cells are the cells for us that help us make antibodies. This helps those plasma cells survive. And we know myeloma is a cancer of plasma cells. And so it turns out BCMA is highly expressed on the myeloma cells as well. And that made it an attractive target. It was not found on most other cells in the body. And so again, around 2015, 2016, there were 4 studies that all started around the same time trying to explore the idea of targeting BCMA with a CAR. The first group was at NCI, the National Cancer Institute. And then we opened a study at Penn. And then Bluebird Biotech and Legend Biotech were 2 small companies that opened multi-center studies. Bluebird was in the US, Legend was in China. And each of these used different CAR constructs. They had slightly different manufacturing. They had a very heavily pretreated patients. But what these early experiences showed us was that this could work. On the far right, you can see ORR, overall response rate, anywhere from 64 to 90% of patients got a response, and then the VGPR, very good partial response, or CR, complete response, was seen in a subset of patients as well. And so this was, again, proof of concept that this could work in myeloma. These patients had really exhausted almost all other options and sort of said, all right, maybe we need to try this in larger numbers of patients. And in fact, the Bluebird product ultimately got partnered with BMS and became ide-cel, or Abecma. And the Legend product was partnered with J&J or Janssen and became cilta-cel, or Carvykti. And this led ultimately to FDA-approved products that we have today to use for our patients. So this was really the sort of background that led ultimately to those being available. And so today we're fortunate to have 2 FDA-approved products where we can use this for our patients. Ide-cel was approved in 2021 and cilta-cel in 2022. They both hit the same target, BCMA, but they are structurally different. They have slightly different manufacturing protocols and some different side effects and efficacy, as I'll go through. And initially, we could only use these in very heavily pretreated patients. They had to have had 4 prior treatment lines that had failed. They had to have had a prior what's called proteasome inhibitor, like Velcade or Kyprolis, an IMiD, like Revlimid or Pomalyst, and a CD38 antibody, like daratumumab or isatuximab. And subsequently, we got updated approvals in 2024 to use these as early as 1 prior treatment line for patients. And so that's currently where we have the ability to give these to patients. They've had at least 1 treatment or 2 treatments, their disease has come back, and now we can possibly give these to patients. And so I'm going to go through some of the data that led to these approvals to give you a sense of sort of what to expect. And so ide-cel was the first one. It was approved based on a study called the KarMMa study. This was a multi-center study, predominantly in the US. 3 different doses of ide-cel were tested. And at the highest dose, 450 million cells, the overall response rate was 82%. So about 8 out of 10 patients got a response, and almost 4 out of 10 got into complete remission. And last year, we saw long-term follow-up of this study. Again, very heavily pretreated patients who really did not have many options. And the median progression-free survival, which is about, think about it as the time you stay in remission, was about 11 months overall. And there was about 5 or 10% of patients that were still alive in remission 5 years out without any other treatment. And the median survival was about 28 months. And this was what led to that initial approval. And there was a follow-up study called KarMMa-3 where ide-cel was compared in a randomized fashion, a phase 3 trial against what's called standard of care. SOC means doctor's choice. You got one of 5 treatments that were available that you could pick for the patients. And these were less heavily pretreated patients. And again, the ide-cel showed a benefit compared to the standard regimens. The median progression-free survival about 13 months versus 4.4 months and higher response rate. So we obviously were very happy about this, but you can see that those curves are still going all the way down. We weren't seeing those potential cures like we were seeing in leukemia or lymphomas. And so we still needed to do better. And so the other CAR product is called cilta-cel, and this was studied in a trial called CARTITUDE-1. And this was a multi-center study that we participated in at Penn. 97 patients were treated with this CAR. And unbelievable, really, response rates when this was first reported. 98% of patients, all but one, actually got into a response. 83% were complete remissions, meaning that we couldn't find the myeloma, and most of those were minimal residual disease negative. And what was just reported this past year was updated data now with 5 years of follow-up, and the median time in remission is about 35 months, so close to 3 years. Again, something that really hadn't been seen or expected in this late-line patient population. And what I think was impressive is that we're starting to see for the first time what's called a plateau on that progression-free survival curve. At 5 years, about 33%, 1 out of 3 of those patients were still alive and in remission with no other treatment, and they got that break from therapy. And that's one of the things that patients say to me when they've had a CAR product. They say, this has been the best sort of time I felt since I started my myeloma treatment, because for the first time I'm not on any therapy. And as you know, most of us, once you start myeloma treatment, you end up staying on it for years. And so this led to a big sort of press release and got an article in the New York Times, and we're saying, we finally started to think about a cure for this cancer. We don't often use the word cure for myeloma. And I think this is still a little premature. I'd like to see longer data to see what happens, and are those patients going to really stay in remission, in that 5 to 10 year part. But for the first time, we're talking about it. We might have the tools, and if we can put them together, as we heard about it in the first talk, and use this with our current treatments, maybe from the start, maybe we'll start to cure a larger proportion of patients. And so, as mentioned, cilta-cel is moving up earlier. We now have a trial called CARTITUDE-4, which compared it to other standard of care regimens called DPD, daratumumab, pomalidomide dex, or bortezomib, pomalidomide dex. These patients had relapsed myeloma, but they only had 1 to 3 prior treatments, not as heavily treated. And again, the CAR T-cell cilta-cel had higher response rates, deeper response rates. They stayed in remission longer, so at 30 months, 60% were still in remission and alive, compared to 26% for the standard control group. And for the first time, we showed a survival advantage, that actually going to the CARs at this time point led to better survival compared to the standard regimens. And so this is what led to the approval in 2024 to use it in less heavily treated patients. And so as we heard, the future of this is now moving up into even the first line. And so you heard about a trial called CARTITUDE-6, which is comparing these CAR T-cells head-to-head with stem cell transplant to see right after you start treatment, you get 4 cycles of Dara VRD, and then you randomize to transplant or CAR T, and then Revlimid maintenance. And that trial is fully accrued, but it's going to be a couple years till we get the readout and really see which one's better, and we have to see about side effects, et cetera. And then there's another trial called CARTITUDE-5 that is not for patients not getting a transplant. They get VRD, Velcade, Revlimid, Dex, and then they're randomized to either CAR T-cells and no other treatment, or Revlimid and Dex as maintenance indefinitely. And so I like that trial because it's really asking a question of fixed duration. Can we just give CARs and stop? Or do we do our standard, which is Revlimid maintenance indefinitely for years? And so that trial also accrued. We're waiting for the readout, and we'll see if that moves these up front. So that's really the good about CAR T cells. But unfortunately, in oncology, as you know, there always are risks and side effects. And so what's the downsides? There's always a trade-off. And the key thing to know is that CAR T cells do have toxicities, and they can be serious and even severe and life-threatening. But we have gotten much better at managing these than when we started this in 2010, when that first patient went to the ICU when we weren't sure what was going on. The most common side effect of CAR T is called cytokine release syndrome, or CRS. And this is when the CAR T cells see the cancer cells, they start to activate and multiply, and it almost feels like the worst flu of your life. It overactivates the immune system. Patients get high fevers, muscle aches, malaise, loss of appetite. And this happens in most patients. We expect it. It typically occurs within the first 7 to 8 days of treatment. And patients are monitored closely. They're often admitted to the hospital at the first sign of a fever, or even the CARs can be given inpatient. And if it's mild, we just treat with Tylenol and fluids and rest. But if it becomes more severe, they start to have any issue with blood pressure, trouble breathing, then we intervene quickly. We give a medicine called tocilizumab, an anti-cytokine drug that blocks this cytokine overactivation and calms down the immune system. And so we're able to manage this, I would say, in 95% of patients, and there still are a small subset that may develop more severe symptoms that do have to go to an ICU for level of support. So you have to be aware of this, but it is something that we jump on quickly and can now manage. And actually, we're giving most of our CARs outpatient now because we expect this and know how to sort of adjust quickly to it. Another side effect is called neurologic toxicity, or neurotox, and there's an early form of this also within the first 7 to 10 days, usually concurrent with the high fevers. Patients can develop confusion, rarely delirium, trouble finding words. Again, it typically responds easily to supportive care or dexamethasone, steroids, but a small subset will have more severe complications. And then there is a later neurotoxicity that can occur anywhere between 2 weeks to maybe 6 to 8 weeks. That can include what's called cranial nerve palsy, something called a Bell's palsy you might have heard of, or a facial droop that you can see sometimes with viruses or other inflammatory states. That tends to be short-lived. With a short course of prednisone, goes away. And more serious, something called Parkinson's-like side effects that can occur in 2 to 4% of patients. That can occur later. And this, I think, we're still trying to understand exactly why people get this. It's a minority of patients. If you pick it up early, it can be reversible, but in some cases it has not been. And so close monitoring for this is really important. We're going to talk more about side effects, I think, in the breakout session, so I'm not going to go through all of these in detail, but we'll definitely have a question and answer session about it. We do think about low blood counts, and some patients need transfusion support early on. Risk of infection, and patients need support for that to support their immune system, something called IVIG, intravenous immune globulin, sort of infusions of good antibodies. And then there have been rare cases of secondary bone marrow cancers and inflammation of the gut. So you need to know about this. This is something we discuss with all of our patients. We talk about the pros and cons and decide whether this is worth it. But I will say, based on the outcomes, particularly for relapsed patients, once the disease has come back a couple of times, we feel that the benefits have far outweighed these risks in terms of disease control, especially compared to other things that are out there for myeloma. And the good news is that we're really just scratching the surface, I think, of CAR T cell therapy. I think these products are wonderful, and we're very glad to have them, but these are sort of the version 1.0 of CAR T cells. And we've learned a lot about how to make these and manufacture them and hopefully make them safer. And so we are currently trying to figure out ways to prevent these toxicities using the current products that are already approved. We're using them earlier so patients have less myeloma tumor burden, because it seems like going into the CARs with a lot of active disease may predispose you to some of these toxicities. And then there are next generation products that are coming out, one called anito-cel, another one called GCF012F, that in early studies seem to have really amazing efficacy, but don't seem to have this neurologic toxicities that we've seen with the early products. And so those are now in phase 3 trials, and we're hoping some of those may get on the market soon as well. There are new CARs that hit other targets, so when you've already had one that hits BCMA and the diseases come back and you go after something else. And then really exciting, I think, are these off-the-shelf and in vivo CAR T cells. And so one of the problems with current CAR T cells is you have to collect the T cells from the patient. It can take sometimes 6 weeks or 8 weeks to get them back. The disease may sometimes progress, or they may have beat up T cells with poor function. And so there are now T cells that can be made from off the shelf from healthy donors that are just frozen, ready to go. And you just order the T cells and give them to the patient. And we're doing a trial with one of these right now, and we'll see how they compare to the autologous CARs made from patients. And then what's really interesting is called in vivo CAR T cells. In this case, instead of trying to do that whole manufacturing process in a test tube for weeks, you just inject the genetic material that encodes for the CAR into the patient, and you make it home to the T cells, and the patient becomes their own manufacturing system. You start to make the CARs in your own blood, and the CARs actually grow and start to kill the tumor cells. And there is a paper just published from China with 4 patients where this was done in myeloma, and it worked. The CARs expanded, and all 4 patients had a response, including 2 complete responses. So it sounds like science fiction, but this is actually where we're going in the future, and some of these products are going to be coming to clinical trials in the US soon. So bright future ahead. I think we're just getting started with this approach, and we need to obviously make it better and safer, but I think we're going to get there, and the hope is that we're going to figure out a way to combine these with our current therapies and really start curing a larger proportion of myeloma patients. So with that, I'd like to just thank my colleagues at our myeloma program, as well as at the Center for Cellular Immunotherapies led by Carl June, who helped lead all of the in-house CAR trials, and thank you for your attention.